All right, this is the second lecture on chapter 24, which has sections on metabolism and nutrition. Here I'm going to talk about lipids and proteins. Um, Remember that lipids and proteins are a type of energy carrying nutrients. These are macronutrients, and that these affect health. Um, Proteins and lipids carry energy, proteins 4 calories per gram, lipids 9 calories per gram. And so we're going to talk about metabolism of these things. These, of course, are used to produce energy, ATP, and that is going to be used by the cell for all activities: transport of molecules into and out of the cell, movement of the cytoskeleton, synthesis of new molecules. ATP in from food intake, ATP is used, um, whether it comes from proteins, carbohydrates, or triglycerides, lipids, doesn't really matter, but it's going to be used for the synthesis of lots of things, for all sorts of energy reactions. Lipids themselves, these are oils and fats. Some lipids are essential, means we have to have them in our diet, but that's relatively few lipids. Lipids in our body are mostly triglycerides. Here's a triglyceride up top here. It has 3 fatty acid chains linked together by a glycerol. Um, we use lots of lipids for structural things like phospholipids for our cell membranes, cholesterol for our cell membranes. We also use some lipids for regulatory things like making estrogen and testosterone, but lipids also serve an important function as an energy source. They are our secondary source of energy, and our best way to store energy long term is in the lipids. When we do use lipids for metabolism, um, we use them as with oxygen only. It requires the use of oxygen. So that means this is going to happen a little bit in the cytoplasm, but mostly in the mitochondria. And so what we do is we separate lipids, the fatty acid parts, from the glycerol part. And then the fatty acids we take apart, and we take them apart with 2 carbon chains at a time, and we convert them to a fatty acid coenzyme A, and we break that down. And that acetyl-CoA is used to enter into the Krebs cycle. And so we break them down 2 molecules at a time. If we have extra lipids or extra energy, we store that as lipids. And so whether it's sugars or lipids, we can convert that to lipid molecules by using acetyl coenzyme A to step by step build up fatty acids. And so this illustration shows you that we can make coenzyme A from a variety of things, and then using coenzyme A, we can link 2 carbon chains onto growing chains, and those things are going to become fatty acids. And so we can make or use lipids, and generally that's okay, that we can do that and it's not a problem. So in our daily life, we can do that. However, if we excessively rely on lipids for energy, okay, we have a problem because we'll produce too much acetyl coenzyme A because we're cutting these things into 2 carbon units. And what'll happen is some of these acetyl-CoA will go through a reaction where they're converted into something else, this beta hydroxybutyrate. And this thing is broken down into what are called ketones. And these ketones, you typically see this in uncontrolled diabetes or no-carb diets. And these accumulate in the blood, and they're pretty strongly acidic, and they're pretty hard to get rid of. We can get rid of them a little bit in our breath and a little bit in urine, but they accumulate and they cause acidosis. a really negative consequence. You're not going to run into this, though, unless you use fats exclusively for energy in the absolute absence of carbohydrates. And so you only see this in that uncontrolled diabetes or in no carb diets. I should also mention lipids are difficult to digest, and the digestion of lipids is slower than any other nutrients. It requires both bile from the liver and lipases. And there's a little bit of lipase in the saliva. There's uh, a little bit of lipase in the stomach, but not really a lot. Most lipase comes from the pancreas, and that's where we do most digestion of lipids. And so, um, and of course, this is slow because lipids don't want to dissolve in fat. In addition, lipids are hard to absorb and hard to transport. And so they wind up being absorbed by lymph vessels that are in the villi called lacteals. And then lipids are combined with proteins, chylomicrons, high density lipoproteins, and low density lipoproteins, so that we can transport them in the blood. And these have implications for things like atherosclerosis, particularly LDLs. So it's not all a great story. Okay, likewise, we can use proteins as a source of energy. These are just chains of amino acids. And amino acids share a lot of similarities. They all have a central carbon attached to a nitrogen or amino group and attached to an acid or carboxyl group. And then they vary in this R group that you see here in yellow. And so there are 20 different types with 20 different R groups. All amino acids contain nitrogen, some contain sulfur. Some are essential. Lots of these are essential. We can't make them, so they have to be in the diet. These have many structural uses, many regulatory uses. Our primary uses of proteins and amino acids are structural and regulatory. That's their main function. They can also be used as a source of energy, but that's not a good source of energy. It's a last resort. And the reason for that is proteins require oxygen to be used, and their different amino acids are broken down different ways, but they all wind up being converted to Krebs cycle intermediates. But before you can do that, you have to strip away urea, the nitrogen group. Here it's shown as NH4, and these NH4s are going to affect blood pH. They're basic, and they're hard to get rid of and very hard on the kidneys. There's also a few amino acids that generate ketones. Okay, so there's really negative things, and don't want to rely on proteins. And we have no big store of proteins. When we have to get rid of proteins, that means we're breaking down cells and tissues to do that for when we need energy. So that happens in starvation. So protein catabolism, that requires oxygen. Most of it is happening in the mitochondria. And these different amino acids are going to enter the Krebs cycle in different places as intermediates. And then some even as are going to send things to the electron transport cycle. Unlike amino acids, proteins aren't hard to digest. Digestion begins in the stomach with hydrochloric acid, HCl, and pepsin. And then it's continued in the small intestine with trypsin and chymotrypsin and peptidases. Again, here we would much rather rely on glucose and glycogen as a source of energy so that we can use proteins as structural and regulatory molecules. Okay, the only time we use proteins are during starvation or when glucose and lipids are available, and this causes wasting, loss of tissue. So a pretty serious problem. Okay, this is less important than knowing carbohydrate catabolism, and both lipids and proteins are feeding into the pathways that we use to do carbohydrates. That's important to know. So we're going to stop there, and there'll be review materials for you to look at later.